MTNR1A and MTNR1B are highly specialized systemic regulators. In the brain's suprachiasmatic nucleus, they govern circadian pacemaking and sleep initiation. In peripheral metabolic organs, like the pancreas, they exert precise control over glucose homeostasis and insulin suppression. Yet these divergent physiological outcomes arise from a biological contradiction. The primary amino acid coding sequence for these melatonin receptors remains identical whether they are embedded in a hypothalamic neuron or a pancreatic beta cell. Standard pharmacological models rely on a highly conserved mechanism for signal transduction. When a ligand binds to a G protein-coupled receptor, or GPCR, it typically engages the GI protein pathway. The GI protein pathway is an intracellular signaling cascade that inhibits the production of cyclic AMP, or CAMP, a secondary messenger molecule that decreases cellular activity. This assumption of rigid structural specificity suggests that a specific receptor structure must produce a uniform cellular response. The reality of melatonin signaling contradicts this. Tissues exhibit distinct pharmacological diversity, raising the question of how a uniform receptor achieves such precise, environment-specific tuning. The receptor's primary genetic sequence alone cannot account for the organism's required tissue-specific functional diversity. Yet another variable dictates the final output. Tissue-specific tuning is achieved through the local expression of independent single transmembrane accessory proteins. These proteins coexist with the main receptor and dictate how it behaves in a specific cellular environment. One of the most critical accessory proteins identified is MRAP2. MRAP2, or melanocortin receptor accessory protein 2, is a single-pass transmembrane protein that acts as a universal modulator for several different receptor families involved in energy homeostasis. The prevailing hypothesis is that MRAP2 physically docks with melatonin receptors at the cell membrane, effectively rewriting their baseline pharmacological rules and ligand sensitivity. To verify this mechanism, we must deconstruct three distinct biochemical assays. By tracing the evidence step by step, we can determine exactly how this rewiring occurs at the molecular level. Moving beyond the primary sequence requires discarding previous assumptions about receptor autonomy. We must first prove physical protein interaction before we can assess any functional output. We establish the baseline physical link using co-immunoprecipitation, or co-IP. Co-IP is a technique used to isolate a specific protein and any of its bound partners from a complex cellular lysate, confirming that MRAP2 and MT-NTNRs can be pulled down together. This extraction method has a specific limitation. It proves the proteins bind somewhere inside the cell, but it does not confirm they interact at the functionally relevant plasma membrane. To prove strict spatial localization, we utilize a bimolecular fluorescence complementation, or BFC, assay. BFC fuses two non-fluorescent fragments to separate target molecules. Venous fragment 1 and venous fragment 2 remain dark in isolation. They emit a signal only if the parent proteins physically dock together to complete the fluorophore structure. The resulting confocal microscopy data is definitive. The fluorescence signal is not scattered throughout the cytoplasm. It is highly localized, strictly bound to the outer boundary of the cell. The spatial data confirms that MRAP2 and melatonin receptors exist as stable, localized heterodimers at the cell surface, operating as a single functional unit. With the physical interaction confirmed, we evaluate the actual signaling output of this new heterodimer, specifically looking at how it alters the GI pathway. We measure this output using a glow sensor CAMP assay. The glow sensor assay is a live cell biosensor system that uses a modified luciferase protein to emit luminescence proportional to real-time changes in intracellular CAMP concentration. This graph plots CAMP luminescence over time. The top line shows a baseline receptor exhibiting naturally leaky CAMP levels. As ratios of MRAP2, 1:1, 1:3, and 1:6 are introduced, a significant drop in baseline luminescence occurs. This gap proves MRAP2 suppresses constitutive activity. Furthermore, we see a distinct shift in the log EC50. Log EC50 is the logarithm of the concentration of a ligand required to induce a half-maximal response from the receptor. MRAP2 alters this value. actively shifting the maximal agonist potency when melatonin is finally introduced. MRAP2 acts as a pharmacological dial. It silences receptor leakiness, locking the structure in an inactive conformation until the ligand arrives, and precisely tunes its sensitivity to incoming melatonin. We must determine whether this altered signaling is driven purely by conformational shape changes, or if MRAP2 is actually regulating how many receptors successfully navigate to the cell surface. To separate these variables, we used fixed-cell ELISA, a plate-based assay that uses antibodies to quantify target proteins on the exterior of intact cells. This chart shows surface expression levels. On the left, as MRAP2 dosage increases, the amount of MTNR1A reaching the cell surface significantly decreases. Contrast this with MTNR1B data on the right. Here, an increasing MRAP2 dosage drives a massive increase in surface display. For MTNR1A, MRAP2 acts as an anterograde block. It physically traps the receptor within the endoplasmic reticulum, preventing it from deploying. For MTNR1B, MRAP2 serves an entirely different function. It acts as a membrane chaperone, actively facilitating the transport of the receptor out to the functional plasma membrane. MRAP2 operates as a bidirectional trafficking regulator. It is capable of simultaneously hiding one receptor subtype deep within the cell while amplifying the surface presence of another. Zooming out from isolated in vitro assays allows us to evaluate the systemic implications of this bidirectional control for the organism's entire metabolic network. Consider the distinct environments. A hypothalamic neuron relies on MTNR1A to govern sleep cycles. A pancreatic beta cell relies on MTNR1B to suppress insulin during the nocturnal fasting period. The organism does not need to evolve two entirely different receptor genes with varied coding sequences to accomplish these different tasks. It simply varies the local concentration of MRAP2 to adjust the behavior and population of receptors on the surface of each specific cell type. This mechanism uses identical primary sequences as modular hardware. The accessory protein then acts as customizable software, downloading new operating instructions based on the tissue environment. MRAP2 completely resolves the initial paradox. Tissue-specific functional diversity is outsourced to the accessory complex, untethering the cellular phenotype from the primary genetic sequence alone. This bidirectional tuning requires the scientific community to reevaluate how we approach GPCR pharmacology. Studying isolated GPCRs in simplified cell models yields incomplete data when native accessory proteins are omitted from the assay. This has immediate implications for drug design. Targeting the unique tissue-specific accessory complex offers a far higher degree of precision than targeting the highly conserved binding site of the GPCR itself, which often causes off-target effects. Mastery of molecular biology relies on mapping the dynamic, combinatorial accessory networks that dictate actual cellular behavior, rather than focusing on primary sequences alone.